Team number 4, gram-negative cocci. Plan. Gram-negative cocci include Neisseria, Moraxella cataralis, and Veillonella. Members of genus Neisseria are catalase and oxidase positive non-motile aerobic gram-negative diplococci. Two species are pathogenic to human. Neisseria meningitis causes pyogenic meningitis, and Neisseria gonorrhoeae causes gonorrhea. Both differ from each other in various aspects. Taxonomy: family Neisseriaceae, genus Neisseria, species Neisseria meningitis and Neisseria gonorrhoeae. Neisseria meningitis meningococcus are capsulated gram-negative diplococci which adjacent sides flattened, lens-shaped, half-moon-shaped, though it is a commensal is nasopharynx of healthy adolescents. Invasive disease such as meningitis or septicemia can occur in susceptible individuals. Cultivation. On blood agar, blood should be immediately injected into blood culture bottles, brain heart infusion or BHI broth, and incubated overnight. Subcultures are made onto blood agar and colonies grown are processed as described for cerebral spinal fluid later. And nasopharyngeal swabs, pus, or scrapings from rashes should be carried in transport media, such as Stewart's media, and inoculated onto selective media, such as Zaire, Martin medium, or New York City medium, to suppress the growth of normal flora. Biochemical properties. Biochemical tests should be carried out to differentiate meningococci from other commensal Neisseria species. Meningococci are catalase and oxidase positive. They ferment glucose and maltose but not sucrose. This can be tested by using rapid carbohydrate utilization test. Antigenic structure. Based on the antigenic nature of the capsule, meningococci can be typed into 13 serogroups, among which only 5 serogroups A, B, C, Y, and W135 account for the majority of cases of invasive disease. Other capsular serogroups and non-capsulated meningococci, 16% of isolates are not capsulated, commonly colonize the nasopharynx of asymptomatic carriers and are associated with invasive disease. And virulence factors. Automembrane proteins. They are the porine proteins present beneath the capsule embedded in the outer membrane. They are of two types, pore A and pore B. Both show antigenic variability and are responsible for serotyping pore B and the serosubtyping pore A of meningococci. Lipopolysaccharide and endotoxin. Like other gram-negative bacteria, meningococci possess endotoxins in their cell wall. Endotoxin binds to CD14 molecules on host cell surface in occasion with toll-like receptor 4. This in turn activates the endothelial cells by inducing release of various inflammatory mediators, such as tumor necrosis factor, interleukin 6, 8, 10, and plasminogen activity inhibitor 1. Endothelial injury is central to many clinical features of meningococci, such as increased vascular permeability, leading to loss of fluid and shock, intravascular thrombosis, leading to disseminated intravascular coagulation, and myocardial dysfunction. And immunoglobulin A proteases cleave mucosal immunoglobulin A, transferrin binding proteins. They help in uptake of iron from transferrin. Pathogenesis. Humans are the only natural host for meningococci. Most common source of infection is nasopharyngeal carriers, mainly children. Mode of transmission, it is by droplet inhalation, and the portal of entry is nasopharynx, and spread of infection from nasopharynx meningococci reach the meninges either by hematogenous route causing septicemia, most common, or by direct olfactory nerve spread through cribriform plate or through conjunctiva. Clinical manifestation. Asymptomatic colonization is the most common presentation. Various presentations include rashes, a non-blanching rash, petechial or purpuric, develops in more than 80% of cases. Septicemia, it is attributed to endotoxin-induced endothelial injury leading to increased vascular permeability and intravascular thrombosis. Waterhouse Friderichsen syndrome, it is a severe form of fulminant meningococci characterized by large purpuric rashes, purpura fulminans, shock, disseminated intravascular coagulation, and bilateral adrenal hemorrhage and multi-organ failure. Pyogenic meningitis is commonly affect young children 3-5 years of age. Presentation includes fever, vomiting, headache, neck stiffness, similar to any other bacterial meningitis except for the presence of rashes. Chronic meningococci, it occurs rarely and is characterized by repeated episodes of petechial rash, fever, arthritis, and splenomegaly. And the post-meningococcal reactive disease, immune complexes made up of capsular antigens and their antibodies developed from 4 to 10 days later lead to manifestations like arthritis, rash, and iritis, pericarditis, polyserositis, and fever. And laboratory diagnosis, specimen collection, cerebrospinal fluid, blood, and nasopharyngeal swab, cerebral spinal fluid examination. First portion is centrifuged and used for capsular antigen detection by chemical analysis. Increase cerebral spinal fluid pressure, increase protein, and decrease glucose, and gram staining pus cell with gram-negative diplococci lens-shaped. Second portion culture on blood agar, chocolate agar, and the third portion is enriched in brain heart infusion broth, and nasopharyngeal swab culture on Zeyer-Martin medium. Biochemical test oxidase and catalase positive, and ferment glucose and maltose but not sucrose, and serogrouping by latex agglutination test. Serology, antibodies to capsular antigens. ELISA, enzyme linked immunosorbent assay we use, and the molecular diagnosis by multiplex polymerase chain reaction. Important specimens include cerebrospinal fluid, blood and skin scrapings from petechial rashes from cases and the nasopharyngeal swabs from carriers. Specimens are collected in sterile container and transported immediately without any delay, and the CSF should be processed immediately. It should never be refrigerated as suspected antigens of meningitis, such as meningococci and Haemophilus influenzae, may die on refrigeration. And the nasopharyngeal swabs pass of scrapings from rashes should be carried in transport media such as Stewart's medium and inoculated onto selective media such as Zaire-Martin medium or New York City medium to suppress the growth of normal flora. The bacterial examination of CSF is divided onto the following three portions. First portion of CSF is centrifuged. The supernatant is used for capsular antigen detection by latex agglutination test. By chemical analysis, it reveals elevated CSF pressure, increased protein content, and decreased glucose content. The sediment is used for direct gram staining. Meningococci appear as gram-negative diplococci with adjacent sites flattened lens or half moon shaped, present inside the polymorphs, often extracellular also. This presumptive diagnosis helps to start empirical antibiotics. Second portion of CSF, it is directly inoculated onto an enriched media such as blood agar and chocolate agar and incubated for 48 hours at 37 degrees Celsius under 10% of CO2. At 24 hours of incubation, colonies are small, 1 millimeter, and round, convex, gray, non-hemolytic, translucent. And after 48 hours, colonies become large with opaque raised center and thin transparent margin. And meningococci are very delicate and fastidious to not grow in basal media. Third portion of CSF, it is inoculated on the enriched broths such as brain heart infusion broth and incubated overnight till granular turbidity is produced and then subcultured onto blood agar and chocolate agar. Neisseria gonorrhoeae, gonococcus. Neisseria gonorrhoeae is non-capsulated gram-negative kidney-shaped diplococci. It causes gonorrhea, a sexually transmitted infection, and commonly manifests as cervicitis, urethritis, and conjunctivitis. And motionless, the spore doesn't form in the body, forms of delicate capsule and pili present. And cultivation of Neisseria meningitis. Urethral swab in men and cervical swab is in women are the preferred specimens. Vaginal swab is not satisfactory, and the urethral meatus is cleaned with gas soaked in saline. The purulent discharge is expressed out by pressing at the base of the penis and collected directly onto slides or swabs. Darken or rain swabs are preferred as cotton, and alginate swabs are inhibitory to gonococci. In chronic urethritis, as discharge is minimal, prostatic message is done to collect the secretion. Alternatively, the morning drop of secretion may be collected. Transport media. Specimens should be transported immediately. If not possible, then it should be collected in charcoal-coated swabs kept in Stewart's transport media, or alternatively charcoal containing media or amaze media can be used. Currently, various and microscopy gram staining of urethral exudates reveals gram-negative intracellular kidney-shaped diplococci. Gram staining is highly specific and sensitive in symptomatic mean. And biochemical properties biochemical tests should be carried out to differentiate gonorrhoeae from other commensal Neisseria species. Gonorrhoeae are catalase and oxidase positive. They ferment only glucose but not maltose and sucrose. This can be tested by rapid carbohydrate utilization test. And virulence factor of gonorrhoeae. Pili or fimbri. Pili are the principal virulence factor of gonorrhoeae that help in adhesion to host cell and prevent bacteria from phagocytosis. Pili are composed of pilin proteins which are antigenetically distinct, undergo antigenic and phase variations that help in typing of gonorrhoeae. Automembrane proteins, protein 1, this accounts for more than 50% of total outer membrane proteins. They form transmembrane channels which help in exchange of molecules across gonococcal surface. And there are two major serotypes, POR-B 0.1a and POR-B 0.1b serotypes. And the lipo-oligosaccharide, here the lipopolysaccharide is modified, consisting of only lipid A and core oligosaccharide, but lacks of the repeating O side chain, which is seen in other gram-negative bacteria. It has marked endotoxic activity. And microbiological diagnosis in the cervical culture has a sensitivity of 90%, as cervical swabs contain normal flora. Hence, selective media are preferred, such as their Martin medium is chocolate agar added with vancomycin and vancomycin, colistin, and nystatin. It inhibits commensal Neisseria species. Modified New York City medium lysed blood agar and lincomycin, colistin, trimethoprim, and next Martin Lewis medium inoculated media are incubated at 37 degrees Celsius for 48 hours. Colonies on their Martin media are large, 2.5 mm, gray, convex with crenate margin and raised opaque center. In contrast to meningococci, the colonies of gonorrhoeae vary in size with irregular outline. Blood and synovial fluid cultures should be done in suspected cases of DHI on enriched media, such as lysed blood agar, bisaponin, and chocolate agar. And serotyping we use PCR and immunofluorescent reaction.